Hi, hello. Yeah, this is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College, Autonomous, Mahbubnagar. In our previous class, we learned about microsporogenesis and development of male gametophyte. Today, in our class, we will see the structure of the ovule, types of ovules, and what are the different structures that are uh, concerned to the ovule. Okay. Actually, what are ovules and where they are present, and what is the role of the ovule? So yesterday we saw that in a bisexual flower the male reproductive organs are called stamens and the female reproductive organs are called pistils or carpels. Okay, a carpel consists of ovary. See here, you can see the ovary. This is the ovary, this is the style, and this is the stigma. These three structures constitute a carpel or a pistil. It is the female reproductive unit. The carpels may be many, or the pistils may be many. Either they may be apocarpus or they may be fused. Okay, so inside this ovary, you see the locules, ovular locules. These are ovular locules, and inside these ovular locules are present the ovules. So see these white structures you see here, these are the ovules, and they arise from the place called placenta. The origin, the place of origin of these ovules inside the loculus of this ovary is known as placenta. Okay, so uh, in angiosperms you can see the ovary, that is concealed ovules are present. They are present inside the ovary, but in gymnosperms the ovules are naked ovules are seen in gymnosperms. So as a whole, how to define a egg? What is an megasporangium, or what is an egg? The integumented, the integumented megasporangium is known as a ovule. Inside the ovule, you can find the female gametophyte, that is embryo sac. Embryo sac is present within the ovule. Okay, and within the embryo sac is called as a female gametophyte, and within this embryo sac is present the female gamete, that is egg cell. So, uh, how these ovules are formed or developed? Well, let us see. So, one more time, you can observe here the structure of a pistillary carpel. See, this is the stigma, this is the style, and this is the ovary. Within this ovary is present the ovule. This is the ovule here, and it is attached to the ovary uh, with the help of the funiculus. This region is known as placenta. See, from here arises the ovule. So now we will see how these ovules are developed. Due to periclinal divisions in the cells below the outer layer of the placenta, a papilla-like structure is developed. See, this is the papilla-like structure, a small protuberance, a outgrowth. This papilla-like structure is known as a nucellus. Okay, this is known as nucellus. Within this nucellus, one of the cell gets differentiated into archisporial cell, from which the female gametophyte arises. So, see, from the base of this nucellus develops the integuments. This is the inner integument and this is the outer integument. These integuments grow towards the micropylar region, that is towards the upper side, and they completely cover, leaving a small place, a minute place near the tip. Okay, it is known as micropyle. See, these outer and integuments, they grow from the lower side towards the upper side, leaving a narrow minute space. This narrow minute space is known as micropyle. So this is how ovule develops within the ovary. Okay, so as a whole, this whole structure is known as the body of the ovule, and this body of the ovule is attached to the placenta region by a stalk-like structure called funicle or funiculus. Okay, so uh, then here you can see This is the actual structure of the ovule. This is the funiculus. This is the body of the ovule covered with integuments. Okay, so uh, this region is known as calaja, the region where the nucellus, the funiculus, and the integuments meet. See here, this region is known as calaja. Okay, then the stalk here. This is the stalk, and the stalk of the ovule, as already discussed, it is known as funiculus, and this funiculus is attached to the body of the ovule through a notch like structure called hilum. This is the hilum. See, this is the hilum. Okay, so uh, based on the position of this micropylar region, the embryo sac, the calaja, and the funiculus in angiosperms, the ovules are of different types. What are the different types of ovules? Six types of ovules are discovered based on the position of this micropylar region, the embryo sac, and the position of the funiculus. Okay, so the what are they? The orthotropous ovule, anatropous ovule, hemitropous ovule, compylotropous ovule, amphitropous ovule, and succinotropous ovules are the different types of ovules seen. First one is the orthotropous ovule. What is orthotropous ovule and where it is, in which plants it is seen? The orthotropous ovule, it is the most primitive types of the ovules. They are also known as atropous ovules. Atropous ovules. Okay, here you can see that the micropylar region, the embryo sac, the nucellus, and the funiculus all are present in a straight line. So these are known as orthotropous ovules or straight ovules. These are very primitive and are seen in Raceae family, Piperaceae family, Polygonaceae family, and Euryticaceae family. Then the next one is anatropous ovule. Anatropous ovule, here, these ovules are also called as inverted ovules. Inverted ovules. Okay. So here you can observe that the body of the ovule is at 180 degrees to the funiculus. See, this is the funiculus, and body of the ovule makes 180 degrees to the funiculus. The micropylar comes very close to the funiculus, that is it is present beside the funiculus. Here you can see that the micropylar region, the embryo sac, the nucellus, and the calaza, all of them are in straight line except the funiculus. Funiculus is uh, somewhat parallel to this body of the ovule. So as a whole, the ovule is known as inverted ovule, and the body of the ovule makes 180 degrees with the funiculus. Okay, these are known as anatropous ovules or inverted ovules, and these ovules are seen in monocots and archiclamidae and sympetalae. Next, hemitropous ovules. These are also known as hemianatropous ovules. Here, the curvature of the body shows 90 degrees. That is, the body of the ovule is present at right angles to the funiculus. That is, the body is present perpendicular to the funiculus. This is the funiculus, and the body is present perpendicular to it. Here also, you can observe that the micropylar region, the embryo sac, the calaja, all are present in the same straight line except the funiculus. The funicle is perpendicular to this ovular body, making 90 degrees. So this is known as hemitropous ovules, and this is seen in ranunculus and nothoscordium. Okay. Then comes compylotropous ovules. It is also, this ovule also makes curvature. The body of the ovule, you can see here, it makes 90 degrees angle at the initial stages, but due to the presence of a constriction at the micropylar region, it shows extra growth, overgrowth of this micropylar region makes a little bit more curved when compared to hemitropous ovules, and this micropylar region comes close to the funiculus. Here you can observe that the embryo sac is straight, it is not curved. Okay, so this is compylotropous ovule, and it shows curvature a little bit more than that of hemitropous ovule. And the examples are Leguminaceae family and Raceae family. You can see this type of ovules. And the next one is amphidotropous ovules or amphitropous ovules. Here, this is very important it is asked many a times in the entrance examinations. You need to uh, uh, memorize it, or you need to remember the Amphitropus ovule. Here you can see that the body of the ovule is a little bit more curved when compared to this Compylotropus ovule. The curvature is more than 90 degrees and less than 180 degrees. Here, the micropylar region is it is it comes very close to the funicle. See the micropylar region here, it comes very close to the funicle, and here the embryo sac is curved like a horseshoe. So this is known as horseshoe shaped embryo sac. It is very much curved. Whereas in Compylotropus, you can see a straight embryo sac. But here in Amphitropus ovule, the embryo sac is very much curved and it is horseshoe shaped like. So you have to remember this. Okay, Amphitropus ovule shows a horseshoe shaped curvature embryo sac with horseshoe shape. Okay, and this type of ovules are seen in Alispataceae, Butamaceae, and Centrospermae families. Okay, and the last one is Cercinotropous ovules. In this type, you can see the body of the ovule makes a 360 degrees curvature. That means it makes a complete rotation. Okay, complete rotation. That means it makes 360 degrees with the funiculus. Here the funiculus is very long and it acts as a third integument. It completely encircles the body of the ovule, and it makes the body of the ovule is at 360 degrees to the funiculus. Okay, funicle encircles this body as it is a third integument. Okay, so the curvature is 360 degrees. Okay, then this type of ovules are seen in Opuntia and Plumbaginaceae family. Opuntia and Plumbaginaceae family. These are different types of ovules seen in angiosperms based on the position of the micropyle, the nucellus, the callus, and the Funiculus. Okay, so based on the position of these, the ovules are of six types: Orthotropous, Anatropous, Hemitropous, Compylotropous, Amphitropous, and Cercinotropous. So based on the angles, this is the inverted with 180 degrees centigrade, 180 degrees angle, and this is right angle. It shows right angled body with the funiculus. It is 90 degrees. Compylotropous more than 90 degrees. Amphitropous, it is more than 90 and less than 180 degrees. And Cercinotropous, it shows 360 degrees curvature. Okay, so then comes the next part, that is the integuments. See, these are the integuments surrounding the nucellus. Okay, so uh, these ovules are covered with either a single integument or a double integuments. So if two integuments are present, then such type of ovules are called bitegmic ovules. And if only one integument is seen, then such type of ovules are called unitegmic ovules. And the unitegmic ovules are more advanced when compared to the bitegmic ovules. And these are advanced type. Unitegmic ovules are advanced type when compared to the bitegmic ovules. Unitegmic ovules are seen in sympetale, whereas the bitegmic ovules are seen in uh, polypetale. Okay. Then sometimes a third integument is also seen in some plants. Okay, in some plants a third integument is present. This is known as Aryl. The third integument is known as Aryl, and it is an extra integument which grows from the lower side of the um, lower side of the ovule, that is from the hilum region. Okay, then it completely covers up to the micropylar region. This is the third integument called as Aril, and it is seen in almus. Almus. Okay, almus is the example for the Aril. Then one more example is also there, Myristica fragrans. In Myristica fragrans, what happens? This Aril grows from the outside of the inner integument. from the outside of the inner integument. It grows and it completely covers the inner integument. See, like this, it covers. Okay, so this here in Myristica fragrance, we call it as pseudo-aryl or aryloid. Pseudo-aryl or aryloid. As a whole, what is aryl? Aryl is a third integument which covers both the integuments, the inner and the outer integuments. In Albus, it is present outside the outer integument, but in Myristica fragrance, it grows from beneath the inner integument, from the outside of the inner integument. See, like this. Okay, so this is about the aril. And sometimes what happens after the formation of the seed, these integuments convert into seed coats. Sometimes in some families, such as Asclepidaceae, the outer integument, from the outer integument, hair-like structure grows. Okay, throughout the cells which are present on the outer integument, they give rise to a hair-like structure, and these hair-like structure are present even after the formation of the seed, and they help the seed in dispersal. And this type of hair-like structure which is present on the seed is known as coma, and these seeds are called as comose seeds. Comose seeds. Okay, so and this is caruncle. This is also an extra growth which is present on the micropylar region of the seed. And this is the caruncle is the characteristic feature of the family Euphorbiaceae. And you can easily observe this in the resinous communis seeds. You can it is clearly visible in the resinous communis seeds. These are formed from the cell divisions which occur at the micropylar region, nucellar region, outer epidermis of the nucellar region. These outgrowths they develop from the cellular division of the cells present at the nucellar region surrounding the micropyle. Okay. This extra growth is known as caruncle, and it is the characteristic feature of the family Euphorbiaceae. Okay. Then what is the function of this caruncle? In some plants it is useful for dispersal of the seeds. Okay, dispersal of the seeds. The caruncle becomes swollen by absorbing the water, and useful to separate the seed from the placenta. Okay, this absorbs the water and becomes swollen and pushes the seed away from the placenta, thereby helping the seed to uh, get separate from the placenta. Okay, and also it absorbs water and helps in germination also. So this is caruncle. Then next one is endothelium. See, this is the endothelium. This is embryo sac. That here, the this is the inner integument. Embryo sac is closely attached to the inner integument. So the innermost layer of the inner integument. You can see the cursor here. See, this is the innermost layer of the inner integument. Here the cells are larger when compared to the other cells of the integuments. This layer, they are present in a single layer. They are very large with a dense cytoplasm and conspicuous nucleus. Sometimes these cells may possess more than one nuclei. Okay, see, you can see here two nucleus are also present. One or two nuclei may be seen in this layer. Okay, this layer is known as endothelium, and it is nutritive in function. As it is formed from inner region of the integument, it is also known as integumentary tapetum. As we saw that tapetum is the innermost layer of the anther wall, and which provides nutrition to the microsporogenous tissue. In the same way, here also, this is the innermost layer of the tapete, this inner integument. So we call it as um, integumentary tapetum. Okay, then what is its function? The function of the integumentary tapetum is to provide nutrition to the growing or developing embryo sac. Sometimes some of the scientists also think that these endothelium also secretes some enzymes such as diastase, which helps for easy digestion of the food nutrition or food material from the embryo sac. Then the one more structure which is present outside the ovule is obturator. This is also very important. Hmm. This obturator very important in view of the entrance examinations. Okay, so this is the structure which guides the entry of pollen tube into the uh, embryo sac. Okay, so you can see here, this is the pollen tube. Okay, so, and this is the obturator, the micropylar region followed by the obturator. This is the obturator, and it guides the entry of the pollen tube into the ovule, that is through micropylar region. The pollen tube enters into the ovule through micropyle, and it is guided by this structure called obturator. Okay, this obturator grows along with the ovule and forms a hood-like structure over the nucellus. Okay, so how are the cells here? You can easily observe that these cells are a little bit elongated when compared to all other cells, and they look glandular. See, they look glandular and they are elongated. Okay, so the main function of this obturator is to guide the entry of the pollen tube into the micropylar region. Okay, so this is about the obturator, and the next comes micropyle. So here you can see the region between these two integuments is known as micropyle. Sometimes the micropyle is formed by the uh, outer integument alone. Then in such a case, the micropyle is known as exostome. And in the same way, sometimes it forms from the inner integument alone. So in such case, it is known as endostome. But normally, both the integuments helps form the micropyle. See, this is the micropyle which is formed by both these integument, the outer and the inner integument. Such micropyle is known as entire micropyle. Then comes nucellus. So this is the nucellus, the tissue surrounding the embryo sac. This is the embryo sac here, and the surrounding tissue is known as nucellus. Okay, based on the amount of nucellus present, the ovules are of two types in angiosperms. They are crassinucellate ovules and tenunucellate ovules. These crassinucellate ovules have extensive nucellar tissue. Extensive nucellar tissue. That is, the parietal tissue which is present within the nucellus is more in case of crassinucellate, and they are present in polypetalae. They are seen in polypetalae. Whereas in tenunucellate, only a single layer of nucellus is seen, and these are present in sympetalae. So tenunucellate are advanced when compared to crassinucellate ovules. Then sometimes uh, the cells which are present at the micropylar region, the nucellar cells, the from the epidermal region of this nucellus, the cells undergo periclinal divisions and forms a beak-like structure, and that is known as nucellar beak. Nucellar beak. See here, in between these two integuments, the nucellar and epidermis is seen. The cells which are present at the epidermal region of this nucellus, they undergo more divisions, resulting in the formation of a beak-like structure, and this beak-like structure is known as nucellar beak, and it is seen in Euphorbiaceae members. Nucellar beak is seen in Euphorbiaceae members. Okay, so uh, what is the function of this nucellus? It also provides nutrition. So after complete formation of this embryo sac, the nucellus is used. The complete nucellus, it makes use of this nucellus for its nutrition. But sometimes even after the formation of this embryo sac, a few cells may remain. Okay, a little tissue of this nucellus may remain. Such type of uh, nucellus which persists even after the formation of this embryo sac is known as perisperm. Okay, so it is also very important entrance point of view. What is perisperm? The uh, remaining of the nucellus even after formation of this embryo sac. Complete formation of this embryo sac, a few nucellus is remained. It is not used up. Okay, that remaining of this nucellus is known as perisperm. And this is seen in Cyperaceae and Polygonaceae families. Then two more structures, hypostase and epistase. Hypostase is a disc-like, a plate-like structure present below the embryo sac. If it is present below the embryo sac, it is known as hypostase. What is hypostase? A plate-like structure or a disc-like structure present beneath the embryo sac. If it is present beneath the embryo sac, it is known as a hypostase, and it helps the embryo sac not to grow deep into the nucellus. It prevents the embryo sac to grow deep into the nucellus. Okay. Then in the same way, if the disc is present, if the disc or the plate is present at the tip of this embryo sac, then it is known as epistase. Epistase and hypostase are just plate-like structures present either above or below the embryo sac. Okay. So this is today's lesson. We will just revise it very quickly. Okay. So what we have seen, the ovules are present within the locule of the ovary. The point of origin of these ovules is called placenta. So this is the placenta from where these ovules arise. Within these ovules is present the embryo sac. Okay, integumented embryo sac is known as ovule. Okay, so we have seen the development of this new ovule, how they develop. Um, Due to the periclinal divisions in the cells below the outer layer of the placenta. We already said that they arise from placenta. Here, this is the placenta. So, the cells which are present uh, in the outer layer of this placenta, they undergo periclinal divisions and form a papillae-like protuberance. Okay, this is known as nucellus. Slowly, on the nucellus grows integuments. They grow completely, leaving a minute space called micropyle. And the region uh, in between the uh, nucellus and the funicle and the integuments, the place where these three meet is known as calaja, and this is the body of the ovule, and it is attached to the placenta through a stalk-like structure called funicle. Okay, and the place where this funicle attaches to the ovule is known as hilum. Okay, so then we discussed that there are 6 different types of ovules are present in angiosperms based on the position of the micropyle, the chalaza, and the funicle. Okay, they are orthotropous, anatropous, hemitropous, compylotropous, amphitropous, and cercinotropous. Then we also discussed about the extra growths which are present on the ovule or seed. So this is the one of it. Aril, it acts as a third integument, and in Myristica fragrans, it develops from the outside of the inner integument. Okay. Then caruncle. What is caruncle? It's a sponge-like structure which is present at the micropylar region, and it helps for separation of the seed from the placenta. It also helps in germination. Okay. Then endothelium. Endothelium. What is endothelium? It is the nutritive tissue. It is also known as integumentary tapetum, as it provides the nutrition to the growing embryo sac. This is present in the innermost layer of the inner integument, as it is present in the integument and provides nutrition to the growing embryo sac. It is known as integumentary tapetum or endothelium. It is completely used up by this growing embryo sac. Okay. Then comes obturator. This is also an extra growth which develops from the placenta region, and it grows together with the integuments and forms a hood-like structure over the ovule, and it helps or it guides the pollen tube to enter into the ovule. The cells you can see here, they are glandular in appearance. They are enlarged or elongated cells are seen in the obturator. Okay. Then regarding the nucellus, based on the amount of nucellus, the in angiosperms, the ovules are two types that are the crassinucellate and the tenunucellate. In crassinucellate, the amount of a nucellus is massive, and whereas in a tenunucellate, only a single layer or no nucellus is seen. Okay, so uh, the remainings of the nucellus, even after complete formation of the embryo sac, is known as perisperm. Okay, actually, this nucellus is used up uh, by this growing embryo sac. Okay, so this is today's class. Now, next class we will see megasporogenesis and the structure of the uh, embryo sac. In our next class, we will see megasporogenesis as well as the structure of the embryo sac. Okay, thank you.